Okay, so this is chapter 4, part 2. Again, we're talking about cell structure, and our focus really for the rest of the chapter is going to be on eukaryotic cells. And so when you hear eukaryotic cells, I want you to think eukaryotic organisms. So this is the domain eukarya, which includes all plants, all animals, all fungi, and all protists. The common feature between the cells of all of these types of organisms is that they have a membrane-bound nucleus, which is usually about centrally located, and that nucleus is going to house DNA. And in eukaryotic organisms, DNA is going to be linear instead of circular, like we saw in prokaryotic organisms. And so eukaryotic organisms are going to have several linear chromosomes, again, in their nucleus. You're going to learn when we start to talk about cell division that your DNA in a resting non-dividing cell is actually in the form of chromatin, which you can actually see on these figures, chromatin here, and here, chromatin. So that's a term that you're going to want to be familiar with. Chromatin is, again, what your DNA looks like in a resting non-dividing cell. And that's DNA that's kind of diffuse and spread out, not coiled up into those tight chromosomes, but still linear in nature. Okay? And so again, we'll see that a little bit later. So all eukaryotic organisms have eukaryotic cells that will have a membrane-bound nucleus where we can find the genetic information, DNA. They also have numerous other membrane-bound compartments, which we call organelles. And so in this part of the chapter, we're going to be going through some of those organelles. And we will look at both animal as well as plant cells. So that will be our focus. Okay, and we'll talk about structural differences between the two and then shared characteristics. And so we're going to be going through all these parts and pieces that you see labeled here on these figures. Okay, and I just wanted to show you both of these cells at a little bit of higher magnification, right, blown up a little bit. And so again, here, this is going to be our example of a typical animal cell. And again, cells, depending on where they are in an organism and what their function is, they're going to look different. So not all animal cells will look like this cell. But again, all of the basic structures found in all animal cells are here. And so starting from the outermost structure, we've got the plasma membrane that's going to surround all of the internal contents of the cell. And so again, we'll look at that. Look at right here. Something is being, I think, ingested or maybe released, either endocytized or exocytosed. We have a lysosome. We'll talk about what that is. We have the Golgi body. Here's the nucleus with its nucleolus. We have the endoplasmic reticulum right here, studded with ribosomes. We call it the rough endoplasmic reticulum. We have mitochondria, a vacuole. Here's the smooth endoplasmic reticulum. Peroxisomes. And then we have elements of the cytoskeleton, like microtubules, microfilaments, and intermediate filaments. And so again, we're going to talk about all of those. If I take you over to the plant cell, some added features, these plant cells will also have, where's my pointer? A cell wall. So outside of that plasma membrane, this thick green structure is the cell wall. And cell walls can actually vary in thickness. Another structure that we didn't see in animal cells was chloroplasts. Okay, and if you look here, this large vacuole, plants and animals both have vacuoles, but plants are known to have large, what we call central vacuoles. And these are really important to help maintain the shape of the cell. Okay, and something that's missing from plants, really two things: lysosomes and centrosomes. And again, we're going to talk about both of those as well. So these are nice kind of summary pictures. Okay, so let's start with the plasma membrane. So again, both plant and animal cells, and protists and fungi, so all eukaryotic cells, are going to have a plasma membrane. Plants just happen to have something extra outside of that called the cell wall. Animal cells also have an extracellular matrix that we'll actually talk about in one of the later videos for this chapter. But the plasma membrane is pretty much the outer boundary or barrier. Okay, so this is a nice picture of the phospholipid bilayer. Okay, so it's a bilayer of phospholipids. We saw this in chapter 3, and we're going to see this again in the next chapter where we focus on the membrane. But make sure you know that the phospholipids orient themselves in a very particular manner to form a bilayer. What else is in here? So these blue structures, this is a channel protein. Here's another protein right here. Another one. Here's a coiled protein. Okay, so there's going to be phospholipids arranged in a bilayer. There's going to be proteins embedded in it. If it's an animal cell, the plasma membrane will also have cholesterol embedded in that membrane. And then we also see, if we look at the outside, we see carbohydrates attached to lipid molecules. Those are called glycolipids. And if we have carbohydrates attached to a protein, that's called a glycoprotein. Okay, this would be the outside of the cell. And this is going to be the inside of the cell, which is where our cytoplasm is, right? And so what the plasma membrane does is it separates the internal contents of the cell from the surrounding environment. And a key characteristic or feature of the membrane is going to be that it is semi-permeable. And you're going to hear me say that so many times, especially in the next chapter, and when we do our lab on osmosis. So semi-permeable, or we could also say selectively permeable. And so the plasma membrane is going to really regulate, control the passage of molecules into and out of the cell. Okay. Okay, one more slide on the plasma membrane. I just want to make sure that you're aware of this. There are some plasma membranes, for example, in our bodies, that are going to specialize in absorption. And what membranes do that specialize in absorption is they need to create more surface area. And so what they do is they fold themselves, the membrane folds upon itself, to form a structure called a microvilli, or a microvillus, multiple microvilli. Where do we find cells like this? So your small intestine is an organ of absorption. And so if I were to look at your small intestines, I would find something called the brush border. So this is going to be the lining here. And if I were to look closely, this is what I would see. So this is a nice cartoon drawing, right? And so the lining of your small intestines would be lined with cells. So here's 3 cells. But if I were to look at the membrane facing the lumen, facing the opening, the open side, right, of the intestine, the cavity, what I would find are these folds in the membrane. And so that's pretty neat. So what happens here again is the membrane is going to get folded. And what that does is it increases the surface area of the membrane, which allows it to absorb more. And so this is another example of form equals function, right? And so again, the shape of these cells, the shape of their membrane is directly correlated to the cell's function. They function in absorption. The more surface area they have, the more absorption that can take place. Okay, so we're going to go on from the membrane. We're going to go right below it, right? And we're going to talk about the cytoplasm and the cytosol, which are actually 2 different things, but you can kind of group them into one. So the cytoplasm is the entire region between the plasma membrane and the nuclear envelope. It's that entire space. So in my picture here of the cell, this would be the plasma membrane, right? And here is the nucleus. So it's everything in between, right? So all of these other organelles are kind of sitting in the cytoplasm. We say that the organelles are found kind of suspended in this region. And really around those organelles, we call the cytoplasm around those organelles, we call the cytosol. It's a little bit more jelly-like, a little bit more viscous rather than liquidy. Okay. If we were to look in the cytoplasm, we would find that it's comprised of about 70 to 80% water. And there will also be proteins and enzymes, organic and inorganic molecules within that cytoplasm. lots of reactions will take place in the cytoplasm of the cell. Okay, let's go to the very center now and work our way out. So the most prominent organelle, which again is usually roughly in the middle of the cell, is going to be the nucleus. And the nucleus is going to house our genetic information, so DNA. And again, DNA in eukaryotic organisms is linear as opposed to circular. Okay? And so if we look here, this purple is the nucleus. And you can see that lining the nucleus is the nuclear envelope. And you can actually see that the nuclear envelope is a double membrane. So there are actually two phospholipid bilayers. That membrane is going to be perforated by nuclear pores. And so stuff can come in and out of the nucleus. It just has to get through a nuclear pore complex. And so there's actually proteins in here that are going to regulate the passage of materials. Okay. The fluid in the nucleus is called the nucleoplasm. So just like the cytoplasm, we've got the nucleoplasm. That's going to be the semi-fluid inside the nucleus. You can see these stringy things that are all kind of different colors. That's the DNA. Right now it's in the form of chromatin. So it's DNA plus associated proteins, but not tightly coiled into chromosomes. Instead, kind of diffuse and spread out. Your DNA in this form is accessible. It can be read, and RNA could be made from it. And RNA can then go out into the cytoplasm and make proteins. And so your genes have to be accessible so that they can be expressed. So again, in a non-dividing cell, this is what your DNA will look like. And then here, this dark region, we say it's a dark, dense region, and sometimes there's multiple of these. This is called the nucleolus. And we're going to talk about that. Nope, right here. So I just gave you a brief definition of the nucleolus. Again, it's this dark region. It's prominent in the cell. And what happens there is that's the site of ribosome assembly. And we will talk about ribosomes, but just to give you an idea of what they are, they're actually on the next slide. They're going to be organelles that are responsible for assembling proteins. Okay, and ribosomes are one of those structures that can be found in both prokaryotes and eukaryotes. Okay, in eukaryotes, they're made in the nucleus, inside of that structure called the nucleolus. Okay, so let me give you a little bit more information about ribosomes. So again, they are synthesized in the nucleolus, and then they are shipped out to the cytoplasm. That's where they do their job. What do they look like? So in this figure here, this orange thing and this light blue thing are the two subunits of the ribosome. Okay, so, and they're named pretty easily. The orange is called the large ribosomal subunit, and the blue thing here is called the small ribosomal subunit. And they have to come together to function. They need each other. The ribosome is made up of proteins as well as what's called rRNA, which is ribosomal RNA. And we talked about RNA already, right? So this is ribonucleic acid. We talked, you said that there was 3 different kinds of RNA. Again, one of them is rRNA. So ribosomal RNA is part of the ribosome. So again, they're assembled in the nucleolus, then they're exported through a nuclear pore into the cytoplasm, and it's in the cytoplasm that they do their job. Ribosomes can either be free in the cytoplasm, or they might be attached to either the nuclear membrane or to the rough endoplasmic reticulum. And we're going to look at the endoplasmic reticulum in the next video. What the ribosome does is it's going to read mRNA. So this is messenger RNA. This is a copy of a gene. Okay. The ribosome will read it, and according to the message, it will build a polypeptide. So these little blue spheres here, these are individual amino acids that the ribosome is putting together according to the message in the mRNA, which is a copy of the gene. Okay. There is a third type of RNA on here called tRNA. And again, I'll just briefly mention it. We'll talk more about this in a later chapter. But the T here stands for transfer. And so what transfer RNA does is it transfers amino acids from the cytoplasm to the ribosome. And so it brings in the appropriate amino acid to grow that polypeptide chain. So eventually, this polypeptide chain will be set free, and it will fold up, and it will form a three-dimensional functioning protein. Okay. So we did the plasma membrane, we did the cytoplasm, we did the nucleus, and we did ribosomes. So now we're going to talk about mitochondria. So again, all eukaryotic cells possess mitochondria. Mitochondria are sometimes called the powerhouse of the cell or the energy factory. And what we're saying with those terms is that this is the site of cellular respiration. So this is where the magic happens. This is where our cells are able to take chemical stored energy, glucose, and transform that into an energy that our cells are capable of using, which is ATP. Okay? Anything that we talk about, pretty much, that we say the cell requires energy to do this, it's usually in the form of ATP, which is the product of this process. I did give you the formula in words. You've seen this before. So glucose plus oxygen is going to yield carbon dioxide, water, and ATP. Okay, the carbon dioxide is a byproduct, not important, but the ATP is really, really important. Okay, so again, this happens, it starts in the cytoplasm and then it finishes up in the mitochondria. And so in this image here, I have a cell, right? And so if I pull out one of these organelles here, this is a mitochondria. It looks kind of like kidney bean shaped. This is also a double membrane organelle. So this is the second organelle that we've seen that has two phospholipid bilayers. Remember, the nucleus was one, and now the mitochondria is the second. So there are two membranes. The inner membrane, so the one on the inside, is actually folded up like this, which we already seen this, right? We saw this in microvilli. So what are these folds for? The more folds you have, the more membrane you have. So it increases surface area. And so it's on this inner membrane where most of the ATP is produced. And so the more membrane you have, the more ATP that can be made. These folds are given a special name. They're called cristae, which is right here. Okay. This is a transmission electron micrograph of a mitochondria where you can actually see the inner membrane folds, the cristae. Pretty neat. I also wanted to make sure that you were aware that mitochondria do have their own ribosomes. So these little purple structures in here, those are all ribosomes. So, and they also have their own DNA. So you can see this small circular piece of DNA, which is pretty neat. Because of that, mitochondria are able to make their own proteins, and they also can replicate on their own. Okay, so when your cells go to divide, the mitochondria will replicate. Okay. Lots of words here on this picture. You've seen this picture already, right? So both plant and animal cells, I've got animal cells here, will contain peroxisomes, and then I'm going to talk really quickly about vesicles and vacuoles. Okay, so peroxisomes, you can see in this figure, are these little green circular structures. And so they're small, relatively small compared to the other organelles in the cell. They're round and they're membrane bound. Okay, these are going to be sites where oxidation reactions occur to break down things like fatty acids and amino acids. These are known to be detoxifying organelles. And so we're actually going to talk about an enzyme found in this organelle. We'll talk about it in the lab. But there is the enzyme called catalase that will be in these organelles, and it's going to be responsible for breaking down hydrogen peroxide, which is a byproduct of these oxidation reactions. Hydrogen peroxide, if it were to spill out into your cells, it could be pretty toxic. So inside of the peroxisome, that enzyme catalase will break down the hydrogen peroxide into water and oxygen, which are obviously harmless. So it is a detoxifying organelle. Okay, it's going to metabolize waste products and detoxify them. Secondly on here are vesicles and vacuoles, and we're actually going to mention these again in the next part of the chapter because they're part of what's called the endomembrane system. So vesicles and vacuoles are other membrane-bound sacs, okay? And so right here is an example of a vacuole. And coming off of the Golgi apparatus right here, these would be vesicles, okay? This is a vesicle. And so they have various roles. They may function in storage, okay? So molecules may be stored inside these vacuoles. They may also function in transport. So when a molecule has to be moved from one side of the cell to another, it may be transported inside of a vesicle. Vesicles the membranes of vesicles can fuse with the plasma membrane and other membranes of the cell. And so that's, we're actually seeing that here. This is being internalized and eventually will probably fuse with the plasma membrane. Some of these vesicles may contain enzymes involved in digestion and degradation. Okay, so again, we're going to visit those again in the next part of the chapter. Just wanted to introduce them to you now. And so now we're going to look at major differences between plant and animal cells, and there's a couple that we'll talk about. So first off, animal cells have these organelles called centrioles, and they form what is called the centrosome. So I'm going to introduce that to you, and then again, when we get later in the chapter, when we talk about the cytoskeleton, we'll actually talk about them a little bit more. Another organelle that animals have that plants don't are lysosomes. And again, I'm going to introduce you to them, and then when we talk about the endomembrane system, we'll talk about them once again. In plants will have a cell wall. Animal cells don't have that structure. Plants also have chloroplasts and a large central vacuole, and both of those things are missing from animal cells. So let's talk about some of those. Okay, so the first, so again, animal cells will have these and plant cells do not. They are called centrioles. Okay, so I'm actually showing you right here. Let me just back up actually one slide and see if this shows it. It does. Okay, so centrioles are going to be part of the centrosome, and that's shown right here. Okay, so that's where we're looking. We don't have that structure in plants. Okay, so if I were to take what I just showed you in that last picture and blew it up, made it look a lot larger, that's what we have here. Okay, so this is the centrosome. These are centrioles, so each one is a centriole, and together they make up something called the centrosome. So let's take a look at this. So each centriole is made up of these hollowed out tube-like structures, which are made of microtubules, which again are part of the cytoskeleton. We're going to talk about them a little bit more in the next part of the chapter. But you'll notice the arrangement. There are these triplets of microtubules all around a single microtubule here in the center. Each centrosome is going to have a pair of centrioles, and the centrioles arrange themselves perpendicular to each other. So it's kind of neat. Okay, and again, if we were to count up these triplets, there would be 9 of them. So each centriole, each cylinder, is going to have that same arrangement. What are they used for? So the centrosome, this doublet of centrioles, is going to be found near the nucleus, and it's thought to be how microtubules in animal cells are organized. During cell division, what will happen is this thing replicates. So now the cell has 2 centrosomes, and those centrosomes will move to opposite sides of the cell, and they are going to organize the microtubules that are involved in pulling DNA to separate sides of the cell during mitosis and meiosis. So you'll see these again when we get to chapters 10 and 11. It's strange. So animal cells have these structures and plant cells do not, and we're not really sure why. And although, we believe that the role of this is to organize microtubules, plant cells don't have these, and yet their microtubules are still organized. So the jury's still out and research is still being done on the exact function of these. Okay, but again, you'll see them again. Okay. Oh, yeah, so here's a little bit more. I already kind of said this, but again, they replicate before cell division, they move to the poles, and they're involved in separating chromosomes during cell division. So again, during mitosis and during meiosis, this structure organizes what's called the spindle, which is made up of microtubules. It's a structure responsible for pulling chromosomes apart. And again, the exact role is still kind of a gray area. We're not 100% sure. Okay, second structure, only found in animal cells, is the lysosome. And I actually studied the lysosome during my graduate work. So this again is an organelle, only found in animal cells, and I like to think of the lysosome as kind of the garbage disposal of the cell. So any cellular debris, any garbage, any used up, worn out organelle, or a microbe that's been ingested by a cell, any of those types of things will end up inside the lysosome. And the lysosome will break whatever gets put in there down to its parts and pieces, and then your cell will actually recycle those parts and pieces. So let's take a look here. This is an electron micrograph actually showing two organelles. One is a peroxisome, and the second is a mitochondria that have become kind of worn out and damaged. They no longer function anymore. And so they end up in this membrane-bound compartment. You can see the membrane around it. And the enzymes inside of this lysosome will just chew these up. And again, the small molecules that result will be used again by your cell. Okay, so here it is in cartoon form. So we've got the lysosome here in purple. Okay, we have two worn out organelles, a mitochondria and a peroxisome. And so the lysosome is going to fuse with this vesicle carrying this garbage. Okay, and these enzymes here, represented by these little blue spheres, are going to become activated, and they're going to digest the parts and pieces. And again, these digested particles will be reused by your cell. So again, worn out organelles, worn out molecules, even stuff from the outside. So even like a bacteria that gets taken up, it'll end up in the lysosome eventually, and the lysosome will break it down. When the lysosome breaks down organelles that are part of the cell, we call that autophagy. So it's the cell eating itself. And that's really important that cells are able to do that. Again, we'll talk about the lysosome again in the next part of the chapter. It is part of the endomembrane system. Okay, so just three structures left. So let's talk about what we find in plants. So again, in animals, we found the centrosome and centrioles, and we found the lysosome. We don't find those in plant cells. In plant cells now, we're going to talk about the cell wall, the central vacuole, and chloroplasts. So the cell wall, again, is shown on the outside of the cell here, and it's that green material, right? And you can see cell wall maintains cell shape. So it's a pretty rigid structure. It's going to cover the entire outside of the cell. It'll be just outside the plasma membrane. And again, it's going to give structural support and shape to the cell. It's made of a structural polysaccharide called cellulose, which we saw back in chapter 3. Okay, so you should be familiar with that. The second structure that's only found in plants is the central vacuole, and you can see this here. The central vacuole is going to be large. It's going to occupy most of the area of the cell. It's filled with what we call cell sap, okay, which is just a watery material. And it's really, really important. It plays a key role in regulating water concentration. And so something that plants like, and we'll see this in chapter 5, is they like to be what's called turgid. And so they like to take on extra water and kind of swell up. The plant cells are not going to burst from that swelling because of the cell wall. The cell wall pushes back on the membrane and keeps it so that it doesn't explode. And plants like to be turgid. They like to have a lot of pressure. So they'll put lots of water in the central vacuole, which again gives rigidity to these cells. When we think about plants, plants don't have bones, right? They don't have structures that help them stand upright. And so the only way really that a plant is going to stand upright is if their plant cells are turgid, right? If they're robust and if there is high amounts of pressure in them. If not, they kind of wilt and tilt over. So that central vacuole is really important in regulating water and structural support. Lastly, we've got chloroplasts. So chloroplasts are going to be the site of photosynthesis. And so photosynthetic organisms like plants are going to have these structures, and they have this process occurring inside of them. And so I gave you the formula for photosynthesis. So water plus carbon dioxide plus sunlight is energy, is going to yield glucose. And so plants will make glucose, right? And oxygen as a byproduct. And this process is happening inside of this. And we're going to spend a whole chapter talking about that process in a little bit more detail. And we'll look at the structure of this as well. Something that's not really shown on this figure is that I want you to know that chloroplasts, just like mitochondria, have their own ribosomes as well as their own DNA. So they can make their own proteins and they can also replicate. This is the third double membrane organelle. So remember the nucleus, mitochondria, and now the chloroplast is going to be a double membrane organelle. And so you can see that there's this outer membrane and then there's an inner membrane. And there's a space in between called the intermembrane space. Okay, the fluid inside is called the stroma. And then we have these stacks here. A stack is called a granum, and a single one of these disc-shaped structures is called a thylakoid. And again, when we get into photosynthesis, we'll talk more about those structures. So that's it for part 2.